Hi, welcome to Science Hangout from Science Mission. This is our sixth Hangout. We have Dr. David Katz from Emory University is with us. Welcome, Dr. Katz.
Thank you. It's good to be with you.
Uh, Dr. Katz has won several awards over last several years. He has outstanding postdoctoral fellow 1 in 100 mentor award, Emory University School of Medicine. He has the best poster award, Epigenetics in Gordon Conference at Gordon Conference. Best postdoctoral presentation, first place, best poster, Emory University. Second place in best talk, Southeast Regional Society for Developmental Biology. Outstanding Graduate Teaching Award, Howard Hughes Medical Institute Summer Research Fellowship, to name a few. Welcome, Dr. Katz. So can you please give brief introduction of your background, your education, and some of the things you are doing?
So I actually was fortunate enough to start my research career as a high school student. I used to sort of sneak over to the university to do science experiments on the side. I worked at Stony Brook University on Long Island with Dr. Harvey Lyman, and we were studying euglena and some simple experiments with phototaxis. And then as an undergraduate, I went to Washington University in St. Louis. There I sort of developed my interest further into plants, and I studied stress resistance in barley with Dr. David Ho. And although I don't think I really realized it at the time, I was really introduced there to epigenetics because I had a number of professors, Sally Elgin and Eric Richards and Craig Picard, who taught my basic classes, and they were all epigenetics researchers. And I guess some of that must have rubbed off on me. When I went to graduate school at Princeton University, I studied genomic imprinting and really the epigenetic regulation of gene transcription with Shirley Tillman. And there I was fortunate enough to work on a project where we worked on the first ever mammalian chromatin boundary. And worked on the regulation of that chromatin boundary and how it regulates genomic imprinting. And then after that, I actually moved into C. elegans. And I had the idea that I really wanted to use the germline as a model to study how epigenetics is regulated, particularly how histomethylation is regulated, and the consequences to developmental biology. And that brings us to my own lab. In my own lab, I actually am now drawing on both of those experiences, both as a graduate student and as a postdoc. My postdoc, I should say, was with Bill Kelly at Emory University. And now my lab is drawing on both of those experiences, studying both the C. elegans germline and also applying what we learned in the C. elegans germline to the mouse and really developing some neurobiology that's based on the germline epigenetics.
Interesting. And do you have any role models or mentors who helped you in your career so far?
You know, and I look back on it, I had a couple of wonderful teachers back in junior high school and high school, Judy Cohen and Frank Tirano. And obviously my high school research experience, my first research experience when I was a high school student with Dr. Lyman was amazing. He took a lot of time as a professor to help me out when I was only a high school student. And really, that's where I think I sort of caught the bug because it was amazing to try to be able, as a high school student, to be trying to figure out things that other people didn't know. And so he was a really role model to me. And then I would have to say, you know, my graduate and postdoc mentors have been wonderful. Shirley Tillman is a major player in molecular biology. done some wonderful work working on gene regulation and epigenetics.
Mm-hmm.
And then Bill Kelly is a little bit relatively more new, but in really trying to understand how epigenetics regulates dynamic developmental processes. So I've had a series of mentors along the way who have really encouraged me, and each step I've gotten a little bit of boost and has pushed me on my way.
That's interesting. And like, what do you think are the challenges every day you face in addition to doing research and all?
Well, doing research in these days, it's a wonderful time to be doing research because we have a lot of tools and a lot of the fields are relatively mature. So, for example, my lab is studying epigenetics, but we've gotten into neural behavior and neurodegeneration. And there's enough known about those fields that we can actually figure out where our epigenetic piece of the puzzle fits in. So I think it's a great time to be doing science. On the flip side, as you know, it's also a difficult time to be doing science. Funding isn't necessarily maybe as high as it once was. Funding is a little bit more competitive, both to get grants and to get papers published. So there are some challenges as well. But I think on the balance, I think it's a really fun time to do science.
Okay. And how do you spend your spare time? I know you are busy in your lab, but if at all you have some time, how do you spend it? Like, what are your hobbies?
I have twin 8-year-olds, and they take up a lot of my time.
Okay.
And I am trying. I have had a passion to play soccer and to coach soccer, and I'm trying to pass that on to them by coaching them in soccer. So my time outside of the lab, I spend a lot of time on a soccer field coaching my children.
That's good.
Yeah.
Yeah. And can you talk about your lab, your grad student postdocs in your lab, and their work a little bit?
Yeah, my lab is relatively small. I have currently 4 graduate students, 1 undergraduate, and 2 postdocs. So it's a nice size. There's a community. And as I said, we have we're studying 2 model organisms. And so we have projects both in C. elegans and also in mouse. And what's really nice about what we're doing now is that the phenotypes that we're finding in the mouse are informing our C. elegans biology. So for example, the neurodegeneration work that I'm going to talk about today, we are now interested in potentially developing an Alzheimer's drug going back into C. elegans. And then in the C. elegans work inside of my lab, we're also doing some basic biology, trying to understand further players that are in these pathways and trying to understand consequences. So we have C. elegans projects working on developmental biology, but also on aging. And we're trying to use those systems and what we can learn in those systems to also inform the work that we're doing in mouse. And so my lab has, sort of 2 parts to it, but everybody works kind of hand in hand, and there's a lot of interaction. I'm around the lab a lot because I'm not too old, and I've been, not too far removed from doing experiments day to day. So I'm in the lab doing experiments sometimes, and really talking to people all the time about approaches and ideas and what we're doing.
Okay, how do you motivate these grad students? Because it takes a long time to do research and not every experiment works. So they need some boost every now and then. How do you do that?
Yeah, well, we've been very fortunate, since I've become an assistant professor here at Emory University. We've had a lot of success. So, we'll see if that lasts. But I think, to me, the way I motivate my students is the science. Whether or not the experiments are working, I'm constantly talking to them about ideas and about ways that we can approach those ideas, and they give a lot of input. So that the people in my lab are constantly coming up with new ideas and ways to tweak experiments. So it's a pretty dynamic environment. I think anytime you walk into my lab, you'll find a couple of people discussing vigorously their experiments and having debates about should we do this or should we do that. And I think that kind of environment and the science itself. We've been fortunate enough, as I'm going to talk about it in my presentation, 5-6 years ago, we started out studying a very basic problem in the C. elegans germline. And we've taken that all the way to human Alzheimer's patients. So we're actually going to show some science that we did on postmortem samples from human Alzheimer's patients. And so it's been this kind of wild ride in which we've been fortunate enough to take the basic biology and go all the way to really an applied question. And I think the students and postdocs in my lab have really enjoyed that, in that they may not be working on the basic side, or they may not be working on the more applied side, but there's other people are doing that, and so they can see how their work is fitting into the big picture.
Okay, I think we shouldn't waste more time. I think you should go and present your work. That will help everybody to learn more about what is happening in your lab. Please go ahead.
All right, and before I start, again, I would like to thank you, Dr. Sadasvisha, for inviting me. And I think what you're doing at Science Mission is great.
Share from the left side.
All right, can you see the slides?
Yeah.
Okay, so again, my name is David Katz. I'm an assistant professor in the Department of Cell Biology at Emory University. And so I would like to start my presentation with Conrad Waddington's classic description of the epigenetic landscape. At the top of the hill, Waddington depicted the epigenetic landscape as a ball rolling down this steep valley. And at the top of the hill represents sort of an undifferentiated state, and at the bottom of the hill represents a highly differentiated state. And from the time of Waddington in 1956, with this proposal through the current time, the classic understanding of differentiation is that cells proceed pretty linearly from an undifferentiated state into a more highly differentiated state. And this is why Waddington depicted this as a steep valley where the ball can only roll one way. What I would like to talk to you about today is a story about a histone demethylase and how we think that it plays a critical role in two caveats to this one-way pathway from undifferentiated to differentiated, and how we think that these caveats are actually informing us about the role of epigenetics in neural behavior and also in neurodegeneration. So we started this by looking at one of the most fundamental questions in all of biology, and that is what it means to go through the germline. And so what I'm referring to is, in the germline, you have highly differentiated cell types, sperm and egg. We know that they are highly differentiated because they're the only cells that undergo meiosis and are haploid. And we know that this requires the protein products of many, many, many transcripts. In addition, these cells have highly distinct morphologies that allow them to find one another and undergo the fertilization reaction. And yet these two highly differentiated cells have to come together at fertilization to make the totipotent zygote, or the embryo. And so this suggests that there is a high degree of reprogramming that goes on between each generation. And we were interested in this reprogramming, and in particularly what happens to histomethylation during this reprogramming. And so today I want to talk about one particular histone modification. So we know that DNA is wrapped around nucleosomes, and these nucleosomes consist of histones which can be modified. And these modifications can control the packaging of the DNA and the accessibility such that it affects the ability of the genes that are wrapped around these nucleosomes to either be more highly transcribed or less transcribed. And the histone modification that we want to talk about today is dimethylation of lysine 4 on histone H3. And this is always associated with active genes. And so this initially led to the idea that this histone modification plays a role in transcriptional activation. However, there's increasing evidence that this particular modification is acquired both in enhancers and in gene bodies during productive RNA polymerase elongation. And so you may be wondering, if RNA polymerase is elongating and making transcripts, presumably the gene is already on, and so it doesn't really make sense that this modification is truly involved in transcriptional activation. And so instead, what we've proposed is that it may be serving as a sort of epigenetic transcriptional memory. And the way we think about this is similar to what hopefully you all know as the classic fairy tale Hansel and Gretel. So hopefully you're familiar with that fairy tale, and you'll know, if you are, that in the fairy tale, the kids go through the forest and they leave breadcrumbs behind so they can find their way back. And what we think is that RNA polymerase is doing a similar thing, is leaving a trail behind of where it's been. And we think that if that modification can then be stably inherited through cell division, that it could play as a role as an epigenetic transcriptional memory, maintaining transcriptional states through cell division. And this kind of model makes particular sense during development and differentiation. So the idea is that as genes turn on in somatic tissues, they acquire this histomodification, and this plays a role in maintaining the transcriptional patterns as cells divide to become tissues. However, as you may notice, this kind of a model poses a particular problem to the germline, because in the germline, we also have transcription and we also acquire this histomodification. And if this histomodification could help stably maintain the transcription, then it could result in germline genes being expressed in the embryo of the subsequent generation. And so this suggests that there may be a requirement to reprogram these histomodifications and prevent this from happening. And in fact, there's some evidence for this. So if we go back to actually somatic cell nuclear transfer experiments that were done by John Gurdon and colleagues as early as the 1950s, they were experiments done in Xenopus in frog. And what they showed is that if you take a somatic nucleus and you transplant that into an oocyte, that the oocyte has the ability to convert that transplanted nucleus into a cloned embryo. However, what they found over years is that often the cells that they transplanted from, for like in this case endodermal cells, what would happen in the cloned embryos is that these cloned embryos seem to retain a memory of what they used to be. So if you cloned from endodermal cells, you would get an embryo that inappropriately expressed endodermal genes in the embryo. And they showed that this inappropriate transcriptional memory was completely dependent upon lysine 4 of histone H3. And so we think that it's this histomethylation that's providing this memory. And by analogy, we think that when you go through the germline, there may be a requirement to reprogram this. So when we were beginning to think about this question, the Shi lab at Harvard University identified the first ever histone demethylase. It's a gene called LSD1. And so my lab has been particularly focused on this. And we originally worked on this in C. elegans, where there are 3 versions of this gene, SBR5, T08D10.2, and AMX1. And all of that work is published, so I'm just going to summarize that. And then subsequently, what I'm going to talk to you about today is our work in mammals, in mice, looking at outcomes associated with the loss of LSD1. Okay, so as I said, we started these experiments in C. elegans, and again, we were interested in this question of what happens when you go from gametes to embryo, and what happens to histomethylation. And what we showed is that the histone demethylase LSD1 is deposited into the oocyte nucleus, and it's required to reprogram this mark, H3-dimethyl-K4, at fertilization. When you get rid of it, now this histomethylation can be inappropriately propagated to the embryo, and it can result in things like spermatogenesis genes being expressed in the somatic lineages of the subsequent embryo. And so when I started my lab, what I really wanted to do was to ask, is this also true in mammals, and what are the outcomes associated with that? So the first thing we did was to ask whether or not, like in C. elegans, LSD1 is expressed in the oocyte. So here I'm showing you immunofluorescence. LSD1 is in green and DAPI is in red. LSD1 is expressed in the oocyte nucleus, shown here, and also in the surrounding follicle cells. And this is just a single channel showing that LSD1 is highly expressed in the mouse oocyte nucleus. And so we obtained a conditional knockout from Michael Rosenfeld at UC San Diego, and we deleted LSD1 maternally. And what I want to point out about these crosses is we've deleted LSD1 maternally, and then we've crossed it to a wild-type male to create a heterozygous offspring. And what this does is it enables us to look at the result of only lacking LSD1 maternally in the mother's oocyte, because there's a normal copy right after birth, right after fertilization, which should provide normal zygotic transcription. Okay, so we use two different Cree lines. The first one is GDF9 Cree, the second one is Vasa Cree. Both of these are maternally expressed. And for reasons that are not entirely clear, GDF9 Cree gives us a complete deletion, and Vasa Cree gives us mostly complete deletion, but sometimes is a little bit leaky. And that can be seen here on the right. So this is now immunohistochemistry, looking at LSD1 again in the oocyte nucleus. And when we knock it out with GDF9 Cree maternally, we see that there's a complete loss of LSD1, and that's represented by the brown staining, which is no longer there. But when we look at Vasa Cree now, what we see is sort of a partial loss of function. Most of the time, we see that LSD1 is completely gone, as in this oocyte, but sometimes LSD1 a little bit remains. And so this gives us sort of an opportunity to look at outcomes that are associated with a partial loss of reprogramming. But first, let's start with what happens when you completely lose LSD1 maternally in the oocyte. And the results are quite severe. So what happens is that the embryos arrest at the 1 to 2 cell stage. So here I'm showing you control embryos at embryonic day 1.5. These embryos are 1 cell and 2 cell embryos. When we look at when LSD1 has been deleted maternally, we see 1 cell embryos and occasional things that look like they want to be a 2 cell embryo, but really no proper 2 cell embryos. And by embryonic day 2.5, when the control embryos have gone on to form proper morula, then the GDF9 maternally deleted LSD1 are now either still at the 1 cell stage, or most of them have degraded. And this is quantified on the graph to the right. So this is the percent of total embryos at embryonic day 1.5. And we're looking at fragmented embryos, 1 cell embryos, and 2 cell embryos. So as you can see in the control, there's a large number of 2 cell embryos. And in the maternal mutants, we now have an increase in the fragmented embryos at the expense of normal 2 cell embryos. And so this, again, is consistent with the idea that all of these are arresting at the 1 to 2 cell stage. So we wanted to know whether, similar to what we saw in C. elegans, that this embryonic arrest might be due to the failure to reprogram epigenetic transcriptional memory. And so what we did is that we performed RNA-seq on both oocytes and on the mutant versus wild-type 2 cell embryos. So here I'm showing you a cluster dendrogram, which looks at the relative overlap in expression genome-wide in these RNA-seq data sets. And the first thing I want to show is that if you look at wild-type oocytes shown here versus mutant oocytes shown here, they cluster very similarly. So loss of LSD1 in the oocyte doesn't actually affect the transcription in the oocyte very much. When we look now at a wild-type 2 cell embryo, if we follow along here, we see that a wild-type 2 cell embryo is highly distinct from the oocytes. And that's because zygotic transcription has kicked in all of the embryonic genes. However, when we look at now mutant 2 cell embryos, we see that they actually cluster very similar to both mutant and wild-type oocytes. And what that means is that these mutant 2 cell embryos, instead of activating a zygotic program, they're still expressing all of these oocyte genes. So they fail to undergo the gamete to embryo transition. And so we think it's similar to what we showed originally in C. elegans, that LSD1 is required to enable the progression of embryogenesis by removing this epigenetic transcriptional memory associated with the old program and enabling the embryonic program to turn on. Okay, so as I told you before, we have a second maternal Cree line called Vasa Cree. And again, for reasons that are not entirely clear, it's sometimes a little bit leaky. And you can see that here in the oocyte that has a little bit of LSD1 left, this brown staining. And we wanted to know what would happen if you had most reprogramming, but maybe a little bit of reprogramming being compromised. And what we saw is quite interesting. So most of the embryos still arrest at the 1 to 2 cell stage, but occasionally we have animals survive all the way out to birth. And most of those do not survive to adulthood, but a few of them survive to adulthood. And the ones that we have gotten that survive to adulthood, which has only been 10 or 15 mice over a couple of years, we see that they have very severe obsessive compulsive behaviors. So in these videos, what you can see on the left here is a control video, and this mice is playing like normal and eating. And then on the right, what you're going to see is that the mouse in this cage is obsessively just digging all the time. And the mouse in this cage is now what we call food harassing. So it's taking all the food, chewing it up, and actually distributing it all over the cage. What you can see here, this is now a marble-bearing assay in a control. So a normal mouse pretty much doesn't care about the marbles in the cage. But you can see it when LSD1 is partially deleted in the oocyte, these animals now dig obsessively and bury many of the marbles. And finally, if you look at an open field test, a normal mouse will explore. But when you look at these LSD1 hypomorphs, a lot of them just cower in the corner and don't actually explore the open field. And so we want to show quantification of one of those assays. For the sake of time, I'm only going to show one. So this is a marble-bearing assay, which again is indicative of obsessive compulsive behavior. So what we do is we align 20 marbles inside the cage, and then we give the mice 20 minutes or so in order to move around and explore their environment. A normal mouse and the controls in these experiments basically ignore the marbles, and as you can see, they're relatively left alone. And this results in approximately 5 marbles on average being buried. However, in just a short period of time, in 20 minutes, what you can see is when LSD1 has been slightly affected maternally, then these mice now in 20 minutes will dig obsessively and bury almost every one of the marbles. And again, that's quantified here. The average will bury almost every single one of the 20 marbles. And what I want to point out again is that in these animals, we've partially deleted LSD1 in the oocyte so that there's a little bit of maternal left. We've crossed them to a normal wild-type male. So these have normal LSD1 activity from zygote on. And so the phenotypes that I'm showing you, even the adult behavior phenotypes, we can show that they are due to partial lack of LSD1 maternally in the oocyte. And we think this is really exciting because this gives rise to the idea of epivariation or an epimutation. And that is the idea that a slight defect in reprogramming in the maternal oocyte at fertilization will actually give rise to a stable effect that will actually can be inherited all the way in the adult and cause behavioral abnormalities. Okay, so if we return to Waddington, what I told you at the beginning is that the classic view of differentiation is that you start at an undifferentiated state and you proceed linear to a more highly differentiated state. An obvious caveat to that is in the germline when you go from gametes back to the embryo. So you go from highly differentiated state back to an undifferentiated state. And what we've shown is that LSD1 is required to undergo this transition, in particular to erase sort of epigenetic transcriptional memory associated with the old transcriptional pattern. Now, in the second part of my talk, what I would like to talk about is another caveat and another role for LSD1, in which we think that LSD1 actually, once the cells have now fully differentiated, has an additional role, almost the reciprocal role, in which it helps to maintain differentiated states by continuously preventing them from going back to an undifferentiated state. So in the germline, we think that it actually enables you to go back. And in the somatic differentiated states, we actually think that it continuously prevents you from going back. And what we think is interesting about that is that we think that that role may have have a function in Alzheimer's. So I'll show that data to you now. So unlike in C. elegans and Drosophila, where LSD1 is exclusively expressed maternally in the germline, when we started to look in mice, what we saw, and the same thing is true in humans, LSD1 is widely expressed, including now if we look at the brain. So this is an in situ from the Allen Brain Atlas, showing that LSD1 is expressed in all the cells, as far as we can tell, in the brain. And we're going to be zooming in on this region, which is the hippocampus. This is a series of neurons that are involved in learning and memory. So here on the larger immunofluorescence, LSD1 is shown in red, and a marker for neuronal nuclei is shown in green. And you can see by the colocalization, the yellow, that LSD1 continues to be expressed in all of these learning and memory neurons, these neurons are terminally differentiated and no longer dividing. And so the question is, what is this histone demethylase still doing in these terminally differentiated cells, which are not changing their transcription and not dividing? And so to get at that question, we inducibly deleted LSD1 in an adult mouse using a tamoxifen-inducible system. And so now what I'm going to show you is a zoom in of that hippocampal region. So on the right, here are those hippocampal neurons. These are the cell bodies shown in blue. And then stained in brown is tau, which stains the axonal projections that innervate this hippocampus. Okay, so this is a control, and so all of these are the hippocampal neurons with their associated axons. When we delete LSD1 in an adult animal, within 6 to 8 weeks, every single one of these neurons will die, and that's shown here. So these neurons, the cell bodies, are now completely condensed. And then the tau, this is again stained in tau. Oops, sorry. Is this now stained with tau? And you can see that the axons are completely lost. So all of the innervation of the hippocampus is completely gone. And we know that all of these neurons have died because they're all tunnel positive. So here in control, we're showing DAPI staining and tunnel in green, and then emerge in the right-hand panel. And obviously there's no apoptosis that's going on in the controls. But in mutants, what we can see is that every single one of these condensed nuclei are tunnel positive, indicating that they are undergoing or have undergone apoptosis. And maybe not surprisingly, this results in a very strong learning and memory defect. So we did this in a number of ways. I'm going to show you one of them, which is the Morris water maze. So this is a large tub of water, and we've trained the mice over 5 days to find a platform that's placed in the water normally. And then on the 6th day, we remove the platform and watch where the mouse goes. So as you can see in the first video, a control mouse, when it's placed in the water, will go right over to where the platform is, because normally they like to get out of the water. And you can see this mouse knows exactly where the platform is supposed to be. In contrast, when we look at these mutants, now what we see is that the mouse, even though it's been trained for 5 days, really doesn't know where the platform is at all. And so it more or less swims around aimlessly. And as I said, again, perhaps this is not surprising because all of the learning and memory neurons we think have died. Okay, and the bottom is just a trace showing that, again, the control spends most of its time in the area where the platform was, and the mutants in which all the neurons have died basically swim around aimlessly. So obviously the neuronal cell death that we see and the learning and memory defect is highly reminiscent of Alzheimer's disease. And so we wanted to know whether or not the neuronal cell death that we see has anything to do with the neuronal cell death that's occurring in the human Alzheimer's patients. So this is a very simplistic overview of Alzheimer's disease. And what we know is that Alzheimer's disease, we think, begins with a series of aggregated proteins. So there's amyloid beta, probably initially, and then phosphorylated tau. And these proteins build up as cytoplasmic aggregates. And ultimately, we think that the patients get dementia because we think that these aggregates cause the neurons to die, although it's not really clear exactly how these protein aggregates cause the neurons to die. And so we wanted to know if the neuronal cell death that we're seeing is at all consistent with what's occurring in the human patients. So we performed RNA-seq on our mouse hippocampus. So this is just showing you the replicates that we've done, 2 controls versus 2 mutants where we've inducibly deleted LSD1. This is hippocampus RNA-seq, showing that we get very consistent results and that when you delete LSD1, it's very different from the controls. There's approximately 281 significantly up genes and 124 significantly down genes. But most importantly, when we compare them to the changes that have been observed in the human patients, we do see a high degree of overlap. So now what I'm showing you is scatter plots. So on the x-axis of these plots is the changes in expression in the mouse hippocampus. On the y-axis is changes in expression that occur in human late onset Alzheimer's disease in the prefrontal cortex. Each one of the dots represents a gene with a direct ortholog between mouse and human. And in the left plot, we're showing a group of genes that are involved in immune activation. And in the right plot, we're showing a set of genes that are involved in oxidative phosphorylation. And these are 2 of the main pathways that have been shown to be affected in these Alzheimer's patients. So as you can see in these immune genes, in the Alzheimer's patients on the y-axis, these immune genes are all up. On the x-axis in our mouse hippocampus, we can see that these genes are also all up, which is why these dots segregate almost exclusively to the top right quadrant. And the reason why some of these dots are shown in red is because these are amongst the most highly significantly upregulated genes in our data set. But this correlation doesn't just occur for genes that are up, it also occurs for genes that are down. So in the human Alzheimer's patients, oxidative phosphorylation genes are down, which is why all the dots are below the horizontal axis here. And then in our mice, likewise, we also see that they're down, which is why all of the dots segregate to this bottom left quadrant. And this is not just true of individual pathways. When we actually look genome-wide, we see a high degree of overlap. So I want to start on the bottom by showing you a comparison to the cerebellum of an Alzheimer, of a series. This is a collected version of Alzheimer's patients. And the cerebellum is a tissue that's relatively unaffected. There's relatively little neuronal cell death. And in this plot, each one of the dots represents any one of the 20,000 or so genes that have a direct ortholog between mouse and human. And as you can see in this bottom plot, the dots are roughly distributed equally amongst the 4 quadrants. However, when you look at the top, when you compare to the prefrontal cortex, which is largely affected in Alzheimer's disease, now what you see is a significant skewing into the top right quadrant and the bottom left quadrant. And what this means is that if a gene goes up in the human Alzheimer's patients, it tends to go up in our mice. And when it goes down in the human Alzheimer's patients, it tends to go down in our mice. And this is pretty remarkable, especially considering we're comparing the hippocampus of the mice to the prefrontal cortex of the human patients. This is a tissue that is basically is distinct. It doesn't have it in the mice. Okay, so we think that loss of LSD1 systematically in adult mice recapitulates many aspects of Alzheimer's disease. Specifically, we see the neuronal cell death in the hippocampus and some, just to a lesser extent, in the cortex. We see a learning and memory defect, and we see genome-wide expression changes that match human patients. So we really wanted to know whether or not this enzyme might be affected in the human patients. So now I'm going to show you some staining from post-mortem human cases. This is, in this first slide, I'm showing you an unaffected control. So in all of the unaffected control cases that we've examined, LSD1 is just expressed in the nucleus. This is seen in the zoom in here. LSD1 is strictly confined to the nucleus, and this makes sense since it's a histone demethylase. However, when we look at affected Alzheimer's cases, we see something very distinct. So as I told you earlier, in these Alzheimer's cases, there's aggregation of the protein tau. So this is an antibody to phosphorylated tau. And what you can see are these things called neurofibrillary tangles, these little comet-like structures. And this is cytoplasmic buildup of aggregated tau in these neurons. Remarkably, when we look at LSD1, now instead of being just confined to the nucleus, what we see is often they appear in what look like these neurofibrillary tangles, these cytoplasmic aggregates of tau. And when we do co-localization, that we can see that phosphorylated tau co-localizes quite nicely with LSD1. And so when we look at a series of patients, what we see is that every single one of the Alzheimer's patients have co-localization of LSD1 with aggregated tau. This occurs at a minimum of 20% of the time and a maximum of maybe 80% of the time. And on average in these human Alzheimer's patients, we see that 50% of the time when you have aggregated neurofibrillary tangles of tau, you also see co-localized aggregated LSD1. Okay, and so this has led us to the following idea. We know that LSD1 is continuously required to maintain these neurons. What I didn't tell you earlier is that the neuronal cell death that we see occurs regardless of the time when you delete LSD1. So if you delete in a young adult or a middle-aged adult or an old adult mouse, what you see is always within 6 to 8 weeks, all of the hippocampal neurons will die. So this suggests that LSD1 is continuously required in these neurons to maintain them. And what we think is in an aging brain, when you get buildup of aggregated tau in the cytoplasm, this somehow grabs LSD1 and sequesters it in the cytoplasm. LSD1 is supposed to be in the nucleus, we think, removing histomethylation and repressing genes. And so when it's now sequestered with tau in the cytoplasm, this now results in inappropriate transcription, all of these activated downstream pathways, and the neuronal cell death that we see in Alzheimer's patients. Okay, so we are really intrigued by this idea that LSD1 could play a downstream role in Alzheimer's, but we still haven't really addressed the question of why is a histone demethylase continuously required to prevent neuronal cell death. So as I told you at the beginning of the talk, we have shown that when you go from these highly differentiated gametes, sperm and egg, to the totipotent zygote, LSD1 plays a key role in erasing this epigenetic memory associated with transcription of germline genes and preventing them from being transmitted to the embryo. I didn't have time to talk about it, but both we and others have shown data in adult stem cell populations, things like neural stem cells and embryonic stem cells, and hematopoietic stem cells. And in those cases, LSD1 also plays a critical role in the stem cell differentiation. So as a stem cell differentiates, it normally has a stem cell transcriptional program, and when those genes are turned on in the stem cell genes, they acquire this histomethylation, H3-dimethyl-K4. And what's been shown by both us and others is that LSD1 is required to erase this methylation during stem cell differentiation. So that such that if you get rid of LSD1 and don't erase this histomethylation and you differentiate the stem cells, now what happens is that they start to differentiate, but they still maintain the expression of stem cell genes. So we know that LSD1 is required during stem cell differentiation to act on these critical stem cell genes and to erase this sort of epigenetic transcriptional memory. And so what we wondered is maybe once those stem cell fully differentiates into something like a neuron, whether LSD1 might then be continuously required to repress the transcription of genes associated with other cell fates, things like stem cell genes that used to be on, and continually prevent them from reactivating by continuously removing this histomodification. And if that's the case, what we would predict is that when we eliminate LSD1, then maybe what we would see is that these stem cell genes come back on. And remarkably, that's what we see. So amongst our top most activated genes in our RNA-seq data set, we noticed a couple of the most critical stem cell genes. So 3 of the 4 Yamanaka factors that are required to reprogram differentiated cells into a stem cell are amongst the top most highly expressed in our RNA-seq data set. So here, if we look at KLF4, so this is a critical stem cell gene, Now we've shown you, we've overlaid both the control in blue and when we deleted LSD1 in the hippocampus in orange. And what you can see is that normally the stem cell gene, of course, is not expressed in hippocampal neurons. And so you just see a background level. But when LSD1 is removed, you can now see robust expression in the RNA-seq. And this is true for KLF4. It's true for c-MYC here. We don't actually see OCT4 in the RNA-seq data set. But then when we go in and look at by immunohistochemistry, what we can see is obviously in control is these stem cell genes are never expressed in hippocampal neurons. But when we look by immunohistochemistry at the protein, you can see in brown here that this stem cell gene KLF4 is now inappropriately being reactivated in a subset of these hippocampal neurons. And that's true here of c-MYC. And we also, even despite the fact that we don't really see much evidence of it in the RNA-seq, we can even see some cases where the critical stem cell gene OCT4 is now re-expressed in every one of these dying hippocampal neurons when LSD1 is gone. So what we think is that LSD1 is playing this role, continuously housekeeping by sweeping the genome and removing these histone modifications at genes that are never supposed to be on. And we think that that's true because, again, when we get rid of it, we can now see the reactivation of these stem cell genes that you're never supposed to be on there. And so, again, if we return to Waddington's model, where the idea is that differentiation is a one-way street. You start at an undifferentiated state, and you go continuously towards more differentiated state. And what we are suggesting is that once you reach this differentiated state, a single histone demethylase is required to continuously maintain the differentiated state of that neuron, for example, by preventing the transcription associated with the undifferentiated state from reactivating. So what we think is that these neurons aren't necessarily so committed to the neuronal cell fate. They are actually continuously preventing themselves from reactivating genes associated with a sort of prior cell fate. And in this way, we think that this continuous requirement is vulnerable to the protein aggregates that are occurring in Alzheimer's disease. So in many ways, what we like to think about or intrigued by is the possibility that Alzheimer's disease is really a disease of the epigenetic loss of cell fate, and that what these aggregates are doing during aging is interfering with this continuous requirement to maintain a differentiated cell fate. Okay, with that, I have to thank all of the people who did the work. The initial story that I told on the maternal role of LSD1 at fertilization was done primarily by a really talented graduate student, Jadiel Wasson, and she had help from a former technician in the lab, Ashley King. All of the neurodegeneration work was done by a really productive collaboration between two graduate students, Dexter Myrick, shown here, and Michael Christopher, shown here. And they collaborated with another graduate student from Jerry Boss's lab, Ben Barwick, who did all the bioinformatics. This is Jadiel Wasson. And I'd like to also thank the following people for collaborations and reagents, particularly Alan Levy, who's helped us with so much of the neurodegeneration work, Victor Fondes, Gary Bissell, Grace Pavlov, Nick Seafried, who've assisted us, Marley Gehring for help with the human patients, Michael Rosenfeld for the mice, Diego Castellan for the Vasa Cree, Jason Schroeder in the behavioral core for helping us with the behavioral assays, and Todd McFarlane, who is a co-author on our eLife paper on the first half of the talk, who did all of the RNA-seq analysis. And finally, the NIH, the NSF, and these Emory funding resources for their funding. Thank you, and I've enjoyed having the time to present.
Thanks, Dr. Katz. Okay, so we have some audience viewers. So if they have any questions, they can type them out. So if no, like I have a couple of things to ask.
Yeah,
it's
sure.
it's a very interesting presentation. I really learned a lot, and it's kind of a step forward in my understanding of Alzheimer's and neuroscience in general.
Thank you.
And one of the questions I have is, you have a on a animal model where memory deficits you see with LSD knockdown.
Yep.
So can you see any tangles or anything in these mice, or it is totally different phenomenon?
So we've looked very hard to see any evidence of protein aggregation, either specifically with beta amyloid or tau, or even more general with things like silver stain, Gaudius. And we don't see any evidence whatsoever of any protein aggregates. Obviously, one possibility was that LSD1 is actually contributing to protein aggregation. And in that case, what we might see is protein aggregates in our mice. And we don't see that. And so what we think is that it's protein aggregation which is interfering with LSD1. And if that's the case, if LSD1 is really downstream, then in our mice where we've just inactivated LSD1, we've basically bypassed the aggregate step and just specifically killed the enzyme. And so we don't really expect to see aggregates in the mice.
Okay. That is interesting. And I was just thinking about, is it a like different phenomenon, like memory and memory loss would be different than tangles, or like is it somewhere it is connected later on, or like I'm a little curious, more curious about how the whole thing is happening.
We
Of
really
course.
think we really think that the memory loss is primarily due to the neurons dying. It's not necessarily clear. I mean, it's possible that the aggregation itself might interfere with the function of neurons before they die. But when you look in the human patients, the human cases of Alzheimer's, what you see, it's actually quite interesting. We don't really know what the dying neurons look like in the human cases, because when you look post-mortemly, what you see is you see neurons that are there, and you see a huge absence of where neurons used to be. But the neurons, we think when they die over many years, they get cleared. And so it just looks like that the density of neurons in those tissues is completely gone. And so it's pretty clear that in the end, many, many neurons die. It's not clear whether it absolutely requires the neurons dying or not for the memories to be gone.
Okay. And I don't see any questions from the audience. Let me have one more questions answered from you. Like the stem cells, what you have mentioned that like in the differentiated form, you need LSD to keep the embryonic transcription of the embryonic genes down.
Yeah.
So I was wondering, can you introduce this LSD1 or increase its expression or like knock it out and see, you can like currently what people are doing is taking a skin cell and modifying into different cell types. Now, can you reverse it back?
You know, we've done some of those experiments. We've tried IPS. What I can say is that if you remove LSD1 and make it easier to activate the stem cell program, the efficiency of IPS goes way up. It happens faster, and you can sometimes get more stem cells formed. When you overexpress it, it actually inhibits the ability to reprogram. So we haven't done as we've tried a little bit of trying to see whether just getting rid of LSD1 alone is enough. We haven't seen that yet. So all those experiments were done where we've either overexpressed or eliminated LSD1 in addition to introducing the Yamanaka factors.
Okay.
But that's consistent with the idea that LSD1 is continuously required in these differentiated cells to repress that program.
Okay. So it's interesting. I think there is a lot of things we could do eventually to learn, unravel what happens with the learning and memory, and you can also sidetrack with the stem cells. I really enjoyed talking, listening to you, and learning more from you. I really appreciate you taking time and present to the audience, and it will be on our website forever, I would think.
I think what you're doing at Science Mission is really great, and I think it's really important that we get the science out there and allow people to have a dialogue about it. So
Yeah.
I thank you for the work that you're doing.
Thank you. And if you have more data to share with the audience, please give me a shout. We can do
Okay.
another hangout whenever you are ready.
Okay.
Thank you very much.
Thank you.